Welcome everybody to our today's colloquium. So to prevent that I'm talking to myself, can you please raise your hand if you can hear me? So if anybody raises the hand, then everybody can. Oh, okay, I see many hands, so seems to work. Let's get started. So it is my great pleasure to present you Kim Nasmyth. So Kim is a real giant of cell biology and biochemistry, and he kindly kindly agreed to present this seminar. And I think this is he presents it from the south of France, if I understood right. So I will give you a very condensed form of Kim's CV, and I will leave out the dozens of awards and distinctions that you can all look up yourself. So Kim is from the UK and studied biology at University of York. So for his PhD, he went to Edinburgh and worked with Murdoch Mitchison and also Paul Nurse on the yeast cell cycle. And the yeast cell cycle is a topic that he would then pursue more or less with little excursions for his whole career. After a short postdoc in Seattle, Kim became a group leader in Cambridge, and where he, among other things, or among many other things, I have to say, cloned the yeast mating type locus. And the yeast mating type locus, I think many of you will know it, it's really something that kept and still keeps dozens of labs busy until to date. And this is definitely a pattern in Kim's career. So whatever he discovers, all the discoveries became major topics, and they are picked up and pursued by hundreds of labs, actually. So 1986, Kim made a very unusual and also very courageous career step because he accepted the offer to be one of the 3 founding senior group leaders of the back then newly founded Institute of Molecular Pathology in Vienna. And one has to say that at this time, Vienna was not really a center for molecular biology, but thanks to Kim, it became one. And Kim stayed at IMP for 18 years, 9 of them as its scientific director. And during this 18 years, the list of the discoveries is really enormous. And I just mentioned 2 very important ones. Kim's lab discovered the anaphase promoting complex and also the molecular mechanism behind sister chromatid cohesion. And I think this is what Kim will also talk about today. So since 2006, Kim is back in the UK, where he heads the biochemistry department of Oxford. And as I said, I don't go through the list of all prizes and distinctions. The most recent prize that speaks for itself was the Breakthrough Prize in the Life Sciences 2018. So before I hand over to Kim, I want to explain the rules of the webinar. So after Kim's talk, we will have 10 minutes for questions. And I ask you to post the questions in the Q&A function of Zoom. So please write them, formulate your question. You can already do that during the talk. And after the talk, I will then read them to Kim and he will answer. So this is much easier than taking the questions directly. So you can, as I said, post your questions during the talk. So now I want to hand over to Kim. Thanks again, Kim, very much for doing this webinars. It's really fantastic that you're joining us, and the screen is yours.
Good. Well, it's a pleasure to be here. We now need to switch. I don't know whether I need to switch. This will stop others. Okay. So can everybody see the presentation? Good, good. Well, it's a pleasure not to be here, but to be engaged with you all. Yes, I was meant to be visiting in April, and unfortunately that didn't happen. But I hope I'll visit the IST physically in the future. Today I'm going to talk about the chromosome structure, particularly with regard to how chromosomes are propagated, namely segregated in mitosis. And one needs to start with an issue that most of you or many people would think was a rather old-fashioned topic is what is the structure of a chromosome? Those of you who've read textbooks over the last few decades would be forgiven for thinking this was a topic that was understood. But in fact, most of the explanations in textbooks for the last 30 to 40 years have been simply stories and bore no resemblance to reality. So let's start with the question of how DNA is woven into a chromosome, not only in mitosis, as you see in this picture, but also in interphase. Now, as you all know, the DNA is packaged into nucleosomes, whose structure you can see on the bottom right-hand side. And really until about 15 years ago, it was thought that really this packaging of DNA into nucleosomes and then higher order structures of nucleosomes interacting with each other somehow miraculously produce chromosomes. This was, quite frankly, this was, people talk about this as a model, but it wasn't a model in any true sense of the word because it never explained anything. And moreover, it didn't explain how, of course, bacteria could have chromosomes whose DNA was highly organized without having any nucleosomes. We now know, largely from work from Tatsuya Hirano and many other colleagues around the world, that in fact taking this tangled schemes of DNA, of which there's vast amounts in any one of our chromosomes, many centimeters in a human, in an axolotl newt it may be 20 times as much as that, and somehow package that into chromatids during mitosis. And we know that this process is driven not by nucleosomes, but by a very different kind of a macromolecular complex, namely the condensing complex. So the nucleosome is shown here, and condensin is shown surrounding that nucleosome. You can see it's a completely different animal. I'll say more about its structure in a moment. And remarkably, Hirano has shown that in fact you can take a xenopus extract and package DNA into chromatid-like structures in the complete absence of nucleosomes. So not only do bacteria do this, but actually eukaryotic cells can do it largely without nucleosomes. Now, many years ago, when we had discovered a related complex related to condensing called Cohesin, of which I will be talking much more later, I started wondering how might Cohesin and condensin function to build chromatids. It was known, of course, condensin is involved in building mitotic chromatids. Cohesin was known to build meiotic chromatids largely during recombination. And essentially, one knew that each chromatid was a cylindrical structure. And it was also known, for example, that condensin accumulated along the longitudinal axis of chromatids. And the question was how would how would a complex with the shape and size of condensin, how might it build chromatids? I just got back from a climbing trip where I'd been climbing in the Dolomites with obviously with ropes, and ropes get tangled up, and you use carabiners to organize them. And secret to good climbing is good rope management, and the secret to good rope management is to make sure your ropes go with carabiners in the right way. So I suggested, after fiddling around with my ropes and carabiners when I got home, that condensin could achieve everything that we knew it had to do by a very simple property, namely the ability to create a small loop of DNA associated with it, and then to extrude that loop through the complex and successively until adjacent complexes met. And as a consequence, you would end up with condensin along the longitudinal axis of the chromosomes. You would end up, if you represent this in 3 dimensions, in a cylindrical structure. And you would also end up with a structure that would hold the axis of the chromosomes together. So this loop extrusion hypothesis seemed to be the only way I could think of at the time of explaining how you build chromosomes. Subsequent work by John Marco and Mirny's lab has elaborated this theoretically, applied it also very specifically to cohesin, and it is now really considered the working hypothesis as to one of the major functions of these types of complexes. Now, a couple of years ago, the Kees Dekker's lab, along with Christian Herring, was able to demonstrate individual, or maybe 1 or 2 condensing complexes actually capable of extruding loops in single molecule experiments, which you see here on the right. I don't want to go into the details of this. The key point is that these loops are extruded very rapidly, up to 1,500 base pairs per second. These experiments obviously done with naked DNA. I think so far nobody's managed to get them to work with nucleosomal DNA. So I would say these are early days, but nevertheless, the condensing complex was shown to have one of the key properties predicted of the loop extrusion hypothesis. Now, chromosomal DNAs must be organized during interphase as well as during mitosis. And it turns out that whereas condensin will organize DNAs into chromatids during mitosis, it's cohesin that organizes DNA during interphase. Now, normally you don't see very much organization in interphase, as we show here on the left. But this organization does exist and has been beautifully revealed in the last 5 years or so using high C techniques, which I won't say anymore. But there is a very dramatic way of observing this organization and showing that it's driven by this cohesin complex, which is related to condensin. If you take an interphase cell and inactivate a regulator of cohesin that is involved in promoting its turnover on chromosomes, if you knock that regulator out, it's a protein called WOPL, then as Jan Michael Peters first showed at the IMP a few years ago, now the cohesin fails. It normally has in interphase, it has a turnover time of somewhere between 5 and 20 minutes. It no longer turns over at all. And now the cohesin organizes the interphase DNA into chromatid-like structures. You see the DNA here, and the cohesin accumulates along the longitudinal axes of those chromatid-like structures. So cohesin is capable of building chromatids in interphase, and condensin will do so in mitosis. And again, more recently, several labs have demonstrated, Mikael's lab and Hung-Tao Yu's lab and Jan Bruger's lab in Dresden have all demonstrated that Cohesin can also extrude loops in vitro using the same sort of techniques as was shown for Condensin. Though these activities can now be measured, there remains a very, very fundamental and crucial question that is totally unresolved, I think, is whether these complexes, or certainly whether Cohesin, extrudes loops as a monomer or as a dimer. If it extrudes loops as a monomer, then you imagine that somehow an individual complex, as shown here, is somehow capable of extruding loops on its own. Namely, it is a dedicated loop extruder. But the possibility exists that in fact that Cohesin and possibly also Condensin might extrude loops as a dimer, in which case it doesn't really need to be a loop extruder as such. They could just be DNA translocases, which if they are held together while they're translocating apart, would then, the net effect of the DNA translocation would extrude loops. And I think this is a fundamental issue that has not been established in the field. Certainly in the case of Cohesin, the extrusion of loops appears to be symmetrical, and it's very hard to imagine how an asymmetric structure, which Cohesin is, and we'll see more when we look at its structure, how it could actually extrude loops symmetrically. So there's a strong argument to suggest that Cohesin might be acting as a dimer. But this is an area which is really not really been well established. Cohesin and Condensin are high related complexes. They are both formed from heterodimers of SMC proteins. In the case of Cohesin, SMC 1 and 3, Condensin 2 and 4. They have a dimerization domain at one end of a long coil. These coils are intramolecular antiparallel. And when the N and C termini attached to those coils come together, they form an ABC like ATPase. And the nature of these ATPases is that ATP will bind to a binding site in 1, and then the phosphates of that ATP will then bind to a particular signature motif on the other head, and vice versa. So those two ABC-like ATPase heads are principally could either be apart in the absence of ATP, or when ATP is bound, the two heads would be engaged and a pair of ATP molecules would be sandwiched between those two heads. More of this later. So the heart of these complexes, these V-shaped SMC heterodimers. Bacteria also have related complexes, but they happen to be homodimeric SMCs. The third subunit that is a so-called Cleiton subunit, the central part of which is largely unstructured, but the N-terminal domain forms an alpha helix that forms a 3 helical bundle with the neck of the coil coming out of the SMC3 ATPase in the case of Cohesin, and the C-terminus forms a winged helical domain that binds to the base of the other ATPase, the SMC1's ATPase. And thereby the Cleiton, by connecting these two ATPase heads, creates a large tripartite ring structure, which we call the SMC Cleiton ring, or SK ring. Just remember that, that's very important, the SK ring. And Condensin has a similar structure, and neither of these trimers, neither of these SK rings on their own have any biological activity. In order for them to do anything, the Cleiton subunit needs to recruit large hook-shaped proteins composed of heat repeats. We'll see structures of them later. And in the case of Condensin, there are 2 of them. They both probably bind DNA, albeit very weakly. They're recruited by the Cohesin, but they don't just bind the Cohesin, they also obviously contact other parts of the complex. In the case of Cohesin, Cohesin is a bit more complicated because as will become apparent, it can do more things than condensin. And in the case of Cohesin, it turns out that there are 3 of these regulatory subunits, and they're called SSE2 or NIPBL, PDS5 or SSE3, or in the mammalian cells, they call them stromal antigens or SA proteins. But I will refer to the yeast names as these were all discovered first in yeast. This is SSE2, stroke NIPBL, PDS5, and SSE3. And at any one time, SSE3 is more or less a permanent member of the complex, and SSE2 and PDS5 compete. So you either have SSE2 there or PDS5 there. Current thinking is that SSE2, which will become clear in my talk, is the protein that activates the ATPase, and that ATPase is fundamental to loop extrusion and DNA translocation. And so the form of Cohesin that would be doing loop extrusion would be SSE2, SSE3 connected to the SK ring. PDS5's role would be larger guide displacing SSE2. It would stop the ATPase and block loop extrusion. PDS5 plays probably a very important role in A, regulating loop extrusion, and B, stabilizing cohesin on chromosomes when it's meant to be holding sister chromatids together. So condensin and cohesin within this large family of SMC Cleiton complexes are first cousins because they share, their regulators are these monophyletic evolutionary related hog-shaped proteins, which are called hawks. And the bacterial proteins, they also have regulators, but they're a different set of proteins, which happen to be called kites. I won't go into the details there. Now, the structural studies of cohesin and condensin are moving very fast now with Cryo-EM. There's huge progress is being made almost on a weekly basis. These are very difficult proteins to study structurally, but Cryo-EM has revolutionized this. And one of the things that has emerged from the Cryo-EM is that two things. One is that the coils of cohesin, for example, are frequently zipped up. These coils, as it were, are interacting with each other over a long distance. And secondly, that there is a region close, about 2/3 the way along the coils, called the elbow, around which the coils bend, fold. So that in fact, you end up the structure on the right-hand side. This is actually a model of a Cryo-EM structure from the Jan Lerver's lab. So this is the dimerization domain, which actually, for historical reasons, is called the hinge. It's nothing to do with being a hinge. Here is the elbow. Here are the coils bent over by the elbow. And then these are the coils coming into the ATPase head domains. And here's the SSE2 subunit. You can see its shape and its size compared to the coils. And this folding seems to be more or less universal feature of SMC protein. Certainly it's true of cohesin and condensin, and also for the mutt beef complex in E. coli. Its biological importance is not understood, but it brings this hinge dimerization domain obviously close to these regulatory subunits bound to the heads and the Cleiton down here. So, for example, one thing, I just want to introduce a technique that has been very important in our lab, both for studying the structure of cohesin in vivo and in vitro. And I'm going to use it here. This is a young student who was an undergraduate in the lab, so the question was, is coil of cohesin folded in this manner, and is the hinge dimerization domain folded onto the coil here in this manner, seen in the cryo-EM structure? Does that actually occur in vivo? And so what Antonio did was he, on the basis of the cryo-EM structure, which was not hugely high resolution, but it was good enough to make some guesses as to what residues within the SMC1 hinge dimerization domain might interact with potential residues in the coil of SMC3. And after rather quickly, he found a pair of residues, particularly this arginine 578 in SMC1 and valine 933 in the SMC3 coil, which when they were mutated to cysteines, he was able then to use this bifunctional thiol-specific cross-linking reagent, which works both in vitro and in vivo, to create this thioether linkages. And if this takes place, then you obviously would be cross-linking SMC1 to SMC3. And what you can see here is yeast cells are viable with these two substitutions. And if you have both substitutions, then about 60 or 65% of the SMC1 is cross-linked to SMC3. This is in living cells when you add BMOE to them. But if you just have one cysteine, then you do not get that cross-link. So that cross-link is undoubtedly taking place between those residues, and therefore this connection really is taking place in vivo. Now I go into this not because this finding is of enormous importance, just to introduce this technique of BMOE-induced cross-linking that depends on the prior substitution of very specific residues picked by structural studies where you inserted cysteines in highly precise positions. Now, cohesin has, though loop extrusion may be a primordial function of all these complexes. The bacterial complexes very probably do this, though it's not been demonstrated in vitro. Cohesin, condensin certainly do it. One suspects the SMC5-6 complex, which is related to them, also does it. But cohesin has presumably at some stage in evolution acquired yet another activity, and this was the activity by which it was first discovered. Namely, it has this function of holding sister chromatids together during mitosis. So these chromatids here, though they are largely separated from each other through the activity of condensin, they still remain connected sufficiently for mitosis to take place. And this sister chromatid cohesion is absolutely fundamental for both meiotic and mitotic chromosome segregation. Because without that cohesion, there is no way that microtubules can ensure that they will pull sister chromatids in opposite directions on the mitotic spindle. So cohesion is absolutely fundamental in resisting the forces exerted by spindles, which thereby creates the tension that stabilizes the connections between microtubules and the kinetochores here. Now you can see this wonderful process. It's one of the most dramatic events of life as cell, of chromosomes lining up in metaphase and then being pulled to opposite poles in anaphase. And it was through the discovery of cohesin and our discovery that the trigger for anaphase, which you see here in this wonderful movie, was in fact the cleavage of the SMC Cleiton ring, the SK ring of cohesin, by an enzyme called separase. Simply clipped in one place the Cleiton subunit, and that is what destroyed sister chromatid cohesion. So this then is what we believe is happening at anaphase. Separase is this very large protease. It's kept inhibited by an inhibitory chaperone. And the anaphase promoting complex, which is this large ubiquitin protein ligase, it activates separase by destroying its inhibitory chaperone. This activates separase, and what it does is it merely clips the Cleiton subunit. And you can actually reproduce this process by replacing separase sites by cleavage sites for the TEV protease, and you can trigger anaphase either in a yeast cell or a Drosophila cell or even in a mouse oocyte by injecting or activating the TEV protease instead of separase. So cleavage of the Cleiton is both necessary and sufficient to trigger the separation of sister chromatids. So the starting point then was how does cohesin hold sister chromatids together? And what is there about the structure that it could explain that, and also could explain why it is that merely cleaving the Cleiton subunit by separase is sufficient to trigger sister chromatid separation. And back in this review I wrote in 2001, when I proposed that cohesin and condensin were loop extruders, I also suggested that cohesin held sister chromatids together by trapping the sister DNAs inside the SMC Cleiton ring. This would explain cohesion. It would also explain how separase destroyed it. And since then, we've really been working on testing this hypothesis and finding out how on earth cohesin does this. And this is the rest of my talk will concern this issue. So cohesin was discovered in my lab and Doug Koshland's lab for yeast genetics. And it's largely still with yeast that we've continued to study. And yeast has the advantages. You can make very small circular chromosomes. They can only be 2 or 3, around 3, as small as 3 kilobases in size. And they can have centromeres on them. They can be segregated. They have sister chromatid cohesion. And we were able to demonstrate that they were indeed, it was cohesin that was holding these sister mini chromosomes together. So the ring model made a very important prediction that if one were to insert cysteines in these 3 interfaces that create the SMC Cleiton ring, namely in the dimerization domain, the hinge dimerization domain between SMCs, between the ATPase heads and the C terminus of the Cleiton, and the other ATPase head and the N terminus. If you inserted cysteine pairs at these interfaces on the basis of known crystal structures or cryo-EM structures, and then were to cross-link those interfaces using BMOE, then if you were to extract the cohesin from the, you break open the cells, immunoprecipitate cohesin, pull down any DNAs associated with them, and then if you were, if you had cross-linked in vivo before you did the IP, then according to the ring model, the sister DNAs, which are circular and are completely decatenated, so topoisomerase 2 has done its business and separated them from each other. It's just cohesin holding them together. Then the DNAs should be trapped in a chemically circularized ring, and you should be able to denature all the proteins in this IP with heating in SDS, and then run these on a gel. And if the ring model is correct, the sister DNAs, which, as I say, are not catenated together in a DNA-DNA catenation, but they would be catenated by this denatured cohesin ring that had been circularized by BMOE. So this is really the technique that we use, is going to be used in almost all the experiments I'm going to talk about henceforth. And so here, again, one makes these pairs of cysteines here, here, and here. And if you have cysteines, all 3 cysteine pairs, then you can get about 20% of the cohesin rings will be chemically circularized. So I mean, each of these is roughly 50%, 50-60% efficiency. So you end up with about 20% circularization. That's a cross-linking in vivo, but you can do the same thing in vitro. So if you take 2 cells, take one yeast cell which has only got 5 cysteines, so you've dropped out one of the cysteines, or you take a cell that's got all 6 cysteines. You do the cross-linking, do the IP, do the SDS, run the gel, and then probe for the mini chromosome. then you can see there's lots of mini chromosomal DNA associated, that was isolated and associated with cohesin, isolated from cells that only had 5 cysteines, but all of it runs as a monomeric supercoil down here. This is just a few nicked circles. But if you've done it from a cell that has all 6 cysteines, and only if it has all 6 cysteines, now you get 2 new forms, one of which is a supercoiled DNA catenated by an individual cohesin ring, and the other is a pair of DNAs catenated by a cohesin ring. And we call these catenated dimers and catenated monomers. And so what we suspect is that, that when cohesin loads onto chromosomes, at least some of the time, it forms these structures. So cohesin is capable of trapping DNAs inside its SK ring, and that is a feature of some of the complexes that are associated with yeast chromosomes. But when you build sister chromatin cohesion during replication, somehow the sister DNAs get trapped inside the same ring, and this is the mechanism by which cohesin holds sisters together. And this finding here is certainly consistent with that hypothesis. We have we've tested a lot of different mutants, and what we have found is that whenever a cell is capable of forming sister chromatic cohesion, then it is also capable of forming catenated dimers. So far as we can tell, catenated dimers, the data is consistent with catenated dimers being synonymous with sister chromatic cohesion. I wouldn't say it's proof, but the data is very, there are no exceptions here. What has become clear is usually when cohesin goes onto chromosomes, it's frequently accompanied by the formation of catenated monomers. But we did find a mutant which was capable of loading onto chromosomes and appears also to be capable of loop extrusion, and yet it was not capable of forming either catenated monomers or catenated dimers. And therefore, we were able to demonstrate that cohesin can load onto chromosomes in yeast and probably can do loop extrusion without entrapment of DNAs inside the cohesin ring. And more recently, Jan-Michael Peters' lab has taken a cohesin ring and cross-linked all 3 interfaces using these similar techniques as I've described here, and shown that that ring is capable of loop extrusion in vitro. So it appears that formation of catenated, that entrapment of DNA inside the Cleiton ring is not necessary for loop extrusion or cohesin's biological activity on chromosomes. But our working hypothesis is that when entrapment of DNAs inside the ring is fundamental to cohesion. In particular, you must entrap sister DNAs inside a single ring. So one of obviously, if we are to make any further progress, we need to start to reproduce this process of entrapment in vitro. And to study, the motivation for studying entrapment is not necessarily to understand loop extrusion, because I've just told you, you probably don't need entrapment for loop extrusion. But the motivation largely is to understand cohesion, which we believe does require entrapment. But as will become apparent that in the course of these studies, I think these entrapment studies possibly shed light into the loop extrusion mechanism as well. So the idea was to take, and this is largely the work of a young graduate student called James Collier with the help of colleagues in Cambridge. You start off with making cohesin trimers, SMC1, 3 Cleiton trimers. They form ring structure, the cohesin, the SK ring, as it were, made in baculovirus and purified to homogeneity. You make the SSC2 and SSC3 Hawk proteins, and you mix them together with circular supercoiled DNA, and you add ATP and you leave them for a length of time, and then you quickly cross-link the 3 interfaces, because you've inserted cysteines in the 3 interfaces here, here, and here. So you've made the 6E version, you cross-link all three interfaces with BMOE. And at that point, you then add SDS and denature all the proteins before you then run them on an agarose gel. And if you have no complex, then the DNA runs as a supercoiled DNA. But if you have SSE2, SSE3, ATP, and you do the cross-linking, and you are a 6Z complex, then now you can see that most of the DNAs are shifted. Either one cohesin ring has entrapped the DNA, 2, 3, and so forth. One fails to resolve them as they get higher and higher. So essentially, we can concatenate most of these DNAs by multiple cohesin rings. And remember, in these experiments, the efficiency of circularization, chemical circularization by BMOE is only, say, 25%. So if there are 3 cohesin rings here, then probably there are 5 times as many. That's 15 before you added the cross-linking reagent. So this is really a quite efficient reaction. Importantly, these catenated forms, the DNA entrapment inside the SK ring. It depends on having all 6 cysteines. You just drop out one of the cysteines if you don't get it, and it's totally dependent upon ATP. So the first thing one would like to know, does it depend on SSE2 and SSE3, which are fundamental to most things that Cohesin does in vivo in yeast? And what you can see here is that here's the reaction in the full reaction with all the proteins, the presence of both SSE2 and SSE3. And you can see the first thing you can notice is actually the reaction is quite slow. It's not really fully completed until about 40 minutes. But crucially, the reaction is totally dependent upon SSE2, and it's totally dependent on SSE3. So you need both SSE2 and SSE3 in order to get DNA entrapment inside the SK ring in this in vitro reaction. This is the input DNA here called I. So what I've referred, so that's very satisfactory. One can get DNAs trapped inside the SK rings, but we know that a very important event in DNA entrapment is the ability to bind. ATP plays a very important role, and indeed the SK entrapment is dependent on ATP's presence and its hydrolysis. Now, as I told you before, ATP will bring those heads together, and we call this state where the heads are held together by a pair of ATP molecules as head engagement. And this is the precursor to its subsequent hydrolysis. And so one suspects that, that head engagement is obviously a key intermediate in the entrapment process, and is presumably a key intermediate in the loop extrusion process. And so we've been able to identify cysteine pairs that would only be cross-linked together with BMOE using when the heads were engaged in the presence of ATP. And so in this state, then you end up taking the SK ring and subdividing it into 2 compartments, either a ES compartment, we call this the SMC, and the head engaged, E for engaged. So engaged heads, SMC, or ES compartment. So you have cysteines here, and you create this subcompartment by cross-linking with BMOE. Or you can make a so-called EK compartment, in which you have cysteine pairs that are specific for engaged heads. And then you have cysteine pairs between the Cleiton and the 2 heads. And so now you can see, you can now create a compartment called the EK compartment, which you can create by cross-linking with BMOE. So now we can ask, do DNAs ever get trapped in this subcompartment or this subcompartment when heads are engaged by using different cysteine pairs? So what you can see is, in fact, that DNAs are very efficiently in this reaction entrapped inside both ES and EK compartments. In fact, this reaction is, as we'll see, is much more rapid than the previous one. And they appear to occur with very similar kinetics. So the reaction is very quick. You get very high levels of entrapment in both within 2 minutes. And remarkably, this entrapment in ES and EK compartments is completely independent of SSC3, which is a vital, and SSC3, remember, is absolutely essential for entrapment in the SK compartment. So this is minus and plus SSC3, minus and plus SSC3, absolutely no effect. This entrapment within ES and EK compartments is at least 20 times more rapid than that within SK rings. So if you look at entrapment after 2 minutes in SK rings with the full reaction in the presence of both SSC2 and 3, then there's virtually none within 2 minutes. And you only get substantial entrapment after many tens of minutes. But both ES and EK, you get very high levels of entrapment. So it's a much more rapid reaction than the entrapment inside the SK ring. Importantly, though it is completely independent of SSC3, it is strictly dependent upon SSC2. So essentially, you get, if you take SSC2 and Cohesin trimers, you'll get DNA very rapidly entrapped in both the ES and the EK compartment. And we assume this entrapment, although we are measuring this with different complexes, because you need distant cysteine pairs to measure entrapment here to here, but we strongly suspect that this entrapment in these two compartments it reflects a single activity, and I'll say what I think that is in a moment. Interestingly, ES and EK compartment entrapment is completely independent hydrolysis. You absolutely need ATP, but you do not need hydrolysis. So these are mutations here that completely abolish hydrolysis. They have no effect whatsoever. So all you need is ATP to bring the heads together, and you need SSC2 and you need that trimer, and the DNA gets trapped in this compartment and this compartment simultaneously. And in this reaction, where you're seeing this simultaneous entrapment in ES and EK compartments, which you see here, here's the input. In the absence of SSC3, this reaction, you can see that there is absolutely no entrapment in the SK ring, okay? So here then, many of you in the audience who are not used to topology, and even some of you who are used to topology, may now at this stage be somewhat confused. How can you get entrapment in this compartment and this compartment, probably simultaneously, without the DNAs being trapped inside the SK ring itself? Well, it turns out there is a very simple explanation. This is not the only one. This is the only one I will consider at the moment. Here you have the SK ring. You have the two heads that are not engaged in the presence of ATP. And the first thing you need to consider is the Cleiton is capable, the heads have got to come apart somehow, and the Cleiton, let's see it sort of as it being folded upwards towards the hinge dimerization domain. You now create a gate through which DNA can pass through those heads. And once the DNA passes through the heads and the heads now engaged in the presence of ATP, you can now see that the DNA, if you then, you will now see that if these heads are engaged like this, the DNA will be trapped in both the ES and the EK compartments. For example, if I have cysteine pairs here and here, you can see the DNA would be trapped in the ES compartment. Or if I have cysteine pairs here, here, and here, this same DNA is entrapped in the EK compartment. But note that in this passage, there has been no opening up of the SK ring itself. None of these three interfaces have opened, and therefore the DNA cannot possibly be in the SK ring itself. So there you have it, a very, very simple explanation for how it is you can get ES and EK entrapment simultaneously in the absence of SK entrapment. And as I say, this reaction is totally dependent on ATP and SSC2 and is very efficient, very rapid. Now, this is where we were a couple of months ago, and then we were basically had got to the stage where we were convinced this is what must be happening. And then appeared, some of you may have seen, there was a paper in Science last month from Hong Tao Yu's lab was published, in which they took Cohesin with a version that couldn't hydrolyze ATP. They added an oligonucleotide, DNA, and they had SSC2 and SSC3, the human orthologs of it. We're only showing the SSC2. And here's part of the structure, the Chromosome structure that they reported. And you can see that what they have here is essentially precisely what we were proposing was occurring when we were measuring ES and EK entrapment. Obviously, in the case of the Chromosome structure, they can't say anything about the topology, either of the Cleiton or of the DNA. But in the case of our reaction, we can say something about both of them. And so therefore these two pieces, and if we therefore believe that in fact when we're measuring this simultaneous entrapment in ES and EK compartments, that what actually we're doing, and this is driven by SSC2, is we're actually creating this structure here by this mechanism that I've shown here. So I think that we don't know that for sure, because our reaction is driven by SSC2 alone. Their structures were created in the presence of both SSC2 and SSC3. But I will put a great deal of money on the fact that SSC2 is forming precisely this structure when you're building this entrapment process here. Well, this hypothesis there are three key findings that are fit with this notion. The first is that in this Chromosome structure, you can see that the DNA is bound on the top of the engaged ATPase heads. These are the ATPase heads. These are the coils. Here's the Cleitons in green. Here's the DNA, and it's bound to residues on top of those heads, but it's also bound to the SSC2. And it turns out that we had previously predicted that this site would be a site of DNA binding, and those residues, when mutated in yeast, cause death, and the cells can't get cohesion onto chromosomes. Here are those four residues there in a higher resolution. So we had suspected that this was where DNA is bound. You are able to show that DNA is an oligonucleotide in a band shift assay is bound, albeit weakly, by the SSC2. But this binding is abolished by mutating these four residues, which are highly conserved between humans and yeast, to glutamate. And if you make those mutations, not only is the yeast cell dead, but also you greatly reduce entrapment in the ES and the EK compartment. You don't eliminate it, but you certainly reduce it. So that's the first key finding that is fully consistent with this hypothesis. The second one is that this hypothesis suggests that the head engagement process is not only driven by ATP, you need ATP to bring these two heads together, but DNA by binding to those engaged heads, and SSC2 by binding both to the coils associated with the heads and to the DNA that is associated with the engaged heads, that all three of those, DNA, SSC2, and ATP would all together be involved in promoting head engagement. And this is the cysteine this BMO-driven cysteine-specific head engagement assay where you're looking at cross-linking. And you can see here that you get efficient cross-linking of the engaged heads in this particular reaction only if you have DNA, SSE2, and ATP present. Now, this is a bit of a cheat because, in fact, to get this result, you've got to leave the magnesium out. You've got to kind of compromise the process. But nevertheless, it demonstrates that these three factors are together bringing the heads together. And so that is certainly consistent. The third key finding is one that we reported several years ago. Namely, if you take the Cohesin trimer, just the SK ring itself, it is completely inactive as an ATPase. You can add DNA, it's still completely inactive. But if you can add SSE2 alone, it's still completely inactive. If you add the trimer and SSE2 and DNA, you now get completely full activity. So basically, the heart of the ATPase is simply the trimer, is SSE2, and the presence of DNA. That's what creates an active ATPase. And so this finding here, our ES-EK entrapment assay and the Chromosome structure all speak to this being an absolutely crucial intermediate in the activity cycle of Cohesin. And what I'd like to suggest, I don't want to speculate as to how Cohesin performs loop extrusion because it's still there's so little is understood. But I just would like to point out that this entrapment of DNA in this site here, in which the DNA is bound to the engaged heads, is probably trapped in this ES-EK compartment, is bound to SSE2, which is part of this and which is now has been witnessed by the Chromosome structures, that this state of DNA trapped inside the ring would be a form of DNA association with the ATP with the ring, which would come and go with the ATPase cycle. And I suggest that this transition would be every time the Cohesin translocates along DNA, it will be transiently forming this structure and then breaking it up. It must be a key feature of this loop extrusion process. So I think we're seeing the heart of the enzymatic machine through this cross-linking studies and from the Chromosome structure. Now, I just want to say one last thing about this in vitro reaction before we move it on into in vivo studies. I told you that you can get this ES-EK entrapment just with SSE2 alone, but if you then add SSE3, and there's this subunit here, this is the actual structure that Hung Tao Yu got. The SSE3 was there, but it's sort of dangling in the wind and probably not doing very much, I suspect, in this particular case. But what we've shown is you've got to have this guy in order to get the DNA into the SK ring itself. Now, we know also from our work and from that of Daniel Panner's lab that SSE3 again binds DNA in this very similar DNA binding groove, similar to the SSE2 groove. You can make a mutate residues involved in that DNA binding. Here's the binding in vitro, and here's the 6E mutation. And it turns out that the SSE3's ability to promote entrapment inside the SK ring is abolished if SSE3 cannot bind DNA. So somehow binding DNA to SSE3 is essential for SK entrapment. And you'll see here in Hung Tao structure that in fact the DNA is not actually bound to SSE3 in his Chromosome structure in the canonical manner. So somehow this complex has sort of got stuck at this stage, and it hasn't gone ahead with some process which would require DNA actually to bind to the SSE3 subunit in this groove here. So what on earth can SSE3 be doing? Now, what I've told you is that SSE2 can form this structure very rapidly, and it does SSE3. So one suspects that when you actually end up with DNA inside the ring, that this would be the starting position. So if this is the starting position with DNA trapped on top of the heads, trapped simultaneously in the ES and the EK compartment with SSE2 also bound here. How could you take this structure and then get the DNA inside the ring? Well, one possibility is that when SSE3 is there, when it combined the DNA, somehow it could open up, for example, the hinge dimerization domain, and DNA then could move through that opened dimerization domain, and then it could shut again, and then the DNA would now be trapped in the SK compartment, but it would have been lost from the ES compartment, but it would now be trapped in the ES compartment, in the SK ring. An alternative is that you create a DNA entry gate not by opening up the hinge dimerization domain, but by opening up a Cleiton SMC head connection and moving DNA through that gap. And that makes a different prediction. So there are two possible mechanisms where you could start off with this state here on the left that's created by SSE2 alone, and then SSE3 somehow opens up either a gate here or a gate here. And in either case, you'd end up with entrapment inside the SK ring. We don't know which of these mechanisms is correct. We need to find out which gate is opened up by SSE3, and this is a very crucial question with regard to understanding how DNAs get trapped inside these rings in vitro. So this work was all done by James Collier, and it was done in collaboration with Byung-Gil Lee in Jan Lover's lab at the LMB. Now, I want my talk to talk about in vivo studies, in principle, to ask how do you actually build cystochromatic cohesion during DNA replication? We know before replication you have DNA entrapped inside cohesin rings. There will also be DNAs that are somehow associated with cohesin that aren't trapped inside those rings, and both of those two populations somehow, presumably, end up being trapped by cohesin rings, or some of them during replication. And a very simple question that as biologists we asked is, if these are the structures that are created during passage of the replosome, where do they come from? Are they derived from cohesin that loads de novo at replication forks, or from cohesin previously associated with DNA, either in this form or some other form, before replication? And so the question, therefore, is can we measure the conversion of cohesin that was associated with DNA before replication? Can we measure the conversion of that cohesin into cohesion after replication? Now, in order to understand these experiments, one important thing one has to realize is that if you express cohesin in a cell, in a G2 cell, that is a cell that's undergone replication, hasn't yet undergone mitosis, the cohesin could load onto the chromosomes, but it won't trap sister DNAs. It will load onto chromosomes, it can form CMs, but it can't form CDs. So here you have a G2 cell, you've got the endogenous cohesin that was present there before S phase, and then during replication, you end up, the DNAs get catenated by this cohesin ring. So that's the endogenous cohesin. But then if you then express in G2 alone a cohesin, then it will go onto chromosomes, it will catenate a single DNA, but it will not catenate both DNAs. And it's very important in these experiments that actually what the form of cohesin that we put on in G2 in this way is not cleavable by separase. So the green cohesin, the endogenous cohesin, is cleavable by separase, but the red one that goes on in G2, it has had the separase cleavage sites mutated, so it can't be cleaved by separase. So the important corollary of this is cohesion is normally only established during S phase. something special happens during S phase. And this is what we're interested in understanding. Now, the other thing, of course, one has to remember is what we're asking is, how do you go from here to here? Do you convert this to here, or do you get other ones coming in de novo? And it's very important to realize that SSE2, which is we've been talking about a great deal, is essential to load cohesin onto chromosomes. So here's cohesin loaded onto chromosomes by a calibrated chipsec, and here's an SSE2 mutant. It can't do it. And as I told you, SSE2 competes with PDS5, and without SSE2, you can't get the ATPase to be active. You can't get cohesin onto chromosomes. Cohesin can't loop extrude. Now, so though SSE2 is essential for loop extrusion, and it's essential to load cohesin onto chromosomes, once you have built sister chromatin cohesion, and in this particular case, we're looking at catenated dimers of these mini chromosomes. So if you take a cell that's undergone replication, and you've got catenated dimers, and then you inactivate SSE2, so you shift a temperature-sensitive mutation from 25 to 37 degrees, wild-type catenated dimers are stable to this temperature shift, and likewise, the catenated dimers made in a temperature-sensitive TS mutant. And the bottom line is that you need SSE2 for all of these activities, but you do not need SSE2 to maintain cohesin on chromosomes, at least in G2 cells. Okay, and that's because under these circumstances, SSE2 is probably replaced by PDS5 that then stabilizes these structures inside cells. Now, so the key question is there a pathway that converts cohesin that's on chromosomes in G1 into cohesion, and that is this somehow converted into this by passage of the replosome? And is this process, does it require the SSE2 to load the cohesin back onto the chromosome, or is it independent of SSE2? Now, obviously, you need SSE2 to get this guy on in the first place, but do you need, once you're on in G1, do you need it to be converted into cohesion during replication? So here's the experiment, and this is quite complicated. I apologize, but there is absolutely no way around it. It is a complicated business. So we go back to these G2 cells. You've got the endogenous cohesin in green, and then you express ectopically in G2 this non-cleavable cohesin. And this non-cleavable cohesin is a 6C cohesin, namely it's got all six cysteines that enable you, therefore, to circularize it with BMOE. All right? Whereas the endogenous one is a perfectly normal cohesin. You then take these G2 cells and allow them to undergo mitosis and enter the next G1 period. During this period, separase will be activated, and the endogenous cohesin will be cleaved by separase, will come off the chromosomes, but the ectopically expressed 6C cohesin will not be cleaved by separase and will remain on the chromosomes. So now you've got cohesin that's been preloaded on the chromosomes in the previous G2, and you now take these G1 cells and allow them to undergo replication, either in the presence of SSE2 or in the absence of SSE2. And you ask, can this population of cohesin that was inherited from the previous cell cycle, is on the chromosomes in G1, can it be converted into catenated dimers, either in the presence or absence of SSE2? And the bottom line is that the answer is yes. In the presence of SSE2, here's cohesin associated with chromosomes, catenated monomers. In the presence of SSE2, you get catenated dimers upon replication. But the same thing happens if you have no SSE2 activity. You had SSE2 active during the G2 period when you loaded the cohesin on, you formed CMs, cohesin was on the chromosomes. You then send these cells replication in the complete absence of SSE2 activity, and you can see this cohesin is converted into CDs. This may appear lower than this, it's just that the loading is different, but the ratios of these different forms are similar. So essentially, cohesin, what this experiment shows is that cohesin that is loaded onto chromosomes before replication can be converted into cohesion that co-entrap sister DNAs or CDs in the complete absence of SSC2 activity. So, and we call this the conversion pathway. We've actually been able to demonstrate this population of cohesin being converted into this population, and this does not require SSC2. You need SSC2 to get those on in the first place, but not to convert them. And somehow, presumably, proteins involved in the replosome may have a role. Of course, there might be a second pathway as well, namely, there might also be a possibility, and we've demonstrated this exists, but of course we haven't excluded the possibility that you could build these structures from cohesin that is loaded de novo during the passage of the replication fork, in which case you might imagine that that reaction would require SSC2. Now, about 12 years ago, the Boone and Brown labs who were working on patterns of synthetic lethality, they and other groups have discovered there are a whole number of non-essential replosome proteins. For example, the CTF4 subunit, CHL1 or DDX11, which is a helicase in mammalian cells, TOF1, CSM3, MRC1, CTF18. None of these replosome components are essential proteins. They're not required for replication per se, but they all lose chromosomes at high rates. And it turned out that the reason why they lose chromosomes at high rates is because they have non-lethal defects in sister chromatid cohesion. And what the Boone and Brown labs showed was that these different proteins fell into two different epistasis groups. One group that contained TOF1, CSM3, CTF4, CHL1. And I apologize, these are the yeast names. They all have different names in mammalian cells. There's timeless tipin, AND1, and DDX11 in mammalian cells. Anyway, that was one group. And the other epistasis group was these components of this alternative form of the RFC complex, which is involved in loading pCNA onto the leading strand. And this MRC1 protein fell into a different epistasis group. And what they showed was that if you combine a mutation in this group with a mutation in this group, you're synthetic lethal. But if you combine mutants in the same group, then there's no synthetic lethality. And so from that, they proposed actually there were maybe two pathways building sister chromatid cohesion, one that involved these proteins and another involved those proteins. But that's where it stood for the next 12 years. It was never clear why you had two pathways and why neither of them was essential. So now we actually have an assay we realize that there are possibilities, there are two possibly different mechanisms for building cohesion. One involves conversion and the other might involve de novo loading. And so now we can ask, are proteins, which of all these proteins might have a role in conversion, very specifically? And so we then take cells which are either wild type or all of these different deletion mutants, none of them are essential genes. And you can load that cohesin on in G2, send them into G1, and then send them through S phase in the absence of these proteins and look for the ability to build CDs from cohesin through the conversion pathway. And what you can see, wild type can do it, MRC1 can do it, mutations in the RFC18, CTF18 proteins can do it just fine, but mutations in CSM3, TOF1, CHL1, CTF4 cannot do it at all. And this is a really very, very remarkable result. It really is absolutely black and white. You do not need any statistics to evaluate these data. No bioinformatics, no hanky-panky No hocus pocus. It's absolutely black and white. Now, so the conclusion is that these set of replicating proteins are essential for the conversion pathway that generates cohesion from complexes previously associated with unreplicated DNAs. And as will become clear, they are in fact also required to maintain cohesin that was on the chromosomes in G1. Something happens when the cells undergo replication. If you don't have the conversion pathway, not only do you not build cohesion, but actually the cohesin that was on there is actually stripped off the chromosomes. And that you can see here is if you do this experiment in the absence of the conversion pathway, so you inactivate CHL1, and here's that G1 cohesin that was built in the previous G2, and then you undergo S phase in the absence of CHL1, in the absence of SEC2, which will allow any de novo loading, and all of this cohesin is completely stripped off the chromosomes when the cells undergo replication. So that's a mini chromosome. You can do the same thing with calibrated chipsex. So this is the level of cohesin along the entire chromosome that was put on in a temperature-sensitive SSC2 mutant in the previous G2. You then send these cells through S phase. In the presence of the conversion pathway, you can see the cohesin remains associated with the chromosome. So basically you have the ratio between what you had before and after replication is roughly 100%. But if you don't have CHL1, the conversion pathway is eliminated, you can see that cohesin, it drops down in level throughout the whole chromosome. So cohesin that's associated with the entire chromosome is stripped off unless you have the conversion pathway. So what seems to be remarkable is this conversion pathway seems to correspond to this epistasis group defined by the Boone and Brown labs. Namely, these proteins are in a single epistasis group because they're all concerned with this conversion pathway. So what about these other proteins? Might they be involved in the de novo loading? It turns out, of course, that if you express cohesin throughout the cell cycle, and so now it can load de novo during replication, and you knock out these genes, they can all form CDs just fine. So the question now is, how do we study the de novo pathway? So what we do is we inactivate the conversion pathway by mutating the CHL1 gene. Okay, so now the conversion pathway doesn't work. And now you can ask, and you express cohesin throughout the cell cycle, for example, and now can you build a cohesion in the absence of the conversion pathway? The answer is you can. And sure enough, that cohesion depends on SSC2. So de novo loading is the cohesion that is built in the absence of the conversion pathway depends on de novo loading. And moreover, having inactivated this pathway is the ability, so you've inactivated this pathway, you can still form CDs because you're getting de novo loading. Is the RFC CTF18 required to build this cohesion in the absence of the conversion pathway? And you can see if you have a temperature-sensitive mutation in CTF8, then now when you inactivate CTF8 and CHL1, you can see you no longer form any CDs. So if you inactivate this pathway and you inactivate this pathway, you no longer form a sister chromatid cohesion or catenated dimers. So the conclusion of this second part of my talk is that there are actually two separate pathways for generated sister chromatid cohesion. There's a conversion pathway. Having loaded cohesin on in the first place, converting cohesin that's on the chromosomes can be performed in the absence of SSC2, so presumably in the absence of the ATPase. But it depends on these replicome proteins, the CTF4, TOF1, CSM3, and CHL1. The position of CHL1 here is very possibly wrong. We don't really know where it exists, but it's a very crucial player because it's rate limiting for this pathway. The second pathway depends on RFC CTF18, probably also on MRC1, and that involves, we suspect, strongly suspect, involves de novo loading of cohesin at the replication fork. And if you can imagine that the de novo loading pathway, you have a very narrow window of opportunity to load cohesin on as the fork goes through. And it turns out that this de novo loading pathway, the SSE2-4 complex, is rate limiting for that. So that is, Tom is trying to tell me to stop, and that is where I am going to stop. We would say in English, the cohesion establishment is a belt and braces job. Namely, you only need a belt or braces. We call them, they're suspenders in America, but we call them braces in English. Neither of these are essential. You need one or the other to hold your trousers up. Some people like to be absolutely certain and have both. And it would appear that the building of sister-chromatin cohesion is a belt and braces job. There are two different pathways. Both can do it. Both are totally conserved. And both seem to play very similar roles, but they're using very different. They're using very different mechanisms. We suspect all of these proteins exist in mammalian cells, and we strongly suspect that both these pathways exist in mammalian cells as well. And this last work of this, the cohesion establishment in vivo, was done almost entirely by Madhu Srinivasan. And it's just been published in eLife, just came out last week. So Tom, thank you very much, and Michael, thank you very much for inviting me to give this talk. I don't know how long I've been far too long probably, but I hope we've still got some time for some questions.
Thank you very much, Kim, fantastic in-depth talk. So while people can now, so everybody can write into the Q&A. If you have specific questions, maybe I start with one. Very general. Actually, isn't it unusual that cohesion does such different things like loop extrusion and sister chromatid adhesion? So isn't it, usually you have specialized complexes for that. So why is it done by one protein, you think? By one complex?
I think it's a very good question, and it's a very bad question. It's a very bad question because you can have the bad is that, you know, remember in biology, never ask the question why. That it's unanswerable. But it's a very good question in that it gets to the heart of the issue, because if you convert it into a how question, is how does cohesion? But it is a fact that it does both. And the question is, how does it do both? What is common between the mechanism of loop extrusion and the mechanism of sister chromatin cohesion? And at present, they seem to be very different sorts of activities. You're absolutely correct to point out this conundrum. Loop extrusion is an active process driven by continually active ATPase, DNA translocase. I think cohesion is a stable structure that is built. It's a one-off thing. It's built at S phase through these two different pathways. Once you build it, you probably no longer require the ATPase. So somehow Cohesin acquired this ability to trap DNAs inside its ring and then utilized it to hold sister chromatids together. But why, I can speculate, but it's, you know, it's unanswerable, really. I think that if I had to guess that condensin and cohesin are highly related, so they're descended from a common ancestor.
Mhm.
The SMC5/6 complex is more related to the bacterial complexes. So that was the original one. And then at some point, either condensin or cohesin evolved. And I would predict that, well, I would predict, but it's not a prediction that will ever be fulfilled, I think, it is that cohesin came first. And the reason for that is that I suspect meiosis came before mitosis. It's very hard to imagine how a cell, you've got a bacterial cell that can segregate its chromosomes just fine. How do you substitute the mitotic system for a system that's perfectly adequate in the bacterium? So clearly, I think that the role of cohesin and in mitosis must have evolved for some other function first, and then was used for mitosis, for cell division. And that other function that is unique to eukaryotic cells, of course, is meiosis and recombination. And cohesin is absolutely central to the whole process of the synaptonemal complex and double-strand break repair. And so I suspect all this cohesin probably evolved in the context of meiosis. And then of course condensin was a derivative. It was a simplification of cohesin, rather than cohesin being a more complicated version of condensin. So I suspect cohesin came first, condensin came second, but that's pure. It's ridiculous to speculate in a way. And I think we have to simply understand, to answer your question, we have to understand how. How do you build cohesion? How do you do loop extrusion? Only then can we begin to speculate in any meaningful sense as to how that might have evolved, how a single complex how one could evolve into the other. But we have to know more about the how, I think, before we answer that question.
Thank you. I now read a question by Franz Klein. What keeps selective pressure to maintain belt and braces? Just that both alone are not sufficient enough? Or is there a differentiation in that one process may normally guide the other? Are in wild type both processes equally efficient?
Well, this is also a very good question. I think Franz partially knows the answer, but he's asking you for all of your benefit. It is remarkable. You knock out either pathway and you end up with very similar cohesion defects. And that implies that both of them are doing a similar degree of lifting. Okay, they're both doing roughly 50%. Now, this is a remarkable coincidence, and this is partly the basis of Franz's question, because usually when you have two proteins have overlapping functions, one of them does the heavy lifting and another one can sort of do it a bit, or vice versa. But these guys are sort of 50-50. And I wonder whether, in fact, the reason why it's 50-50, there may be a functional reason, but it may be fundamental to the mechanism. Namely, if the conversion pathway is operating largely on the maybe say the lagging strand, and the Novo Pathway is working on the leading strand. I mean, you've got these two different strands that emerge, and it could be that they are using they're starting from very different places, one starting from the leading strand and the other starting from the lagging strand. And that's why it's sort of 50-50. Why do they persist? Are there organisms out there that have lost one pathway but not the other? The answer is almost certainly, but I haven't started looking. But what we do know is that at least in yeast, and Franz does know the answer to this question, is that both pathways, though they're neither as essential for mitotic cell division, both are essential for meiosis. And if you mutate either of the pathways, then there is insufficient cohesion between sister centromeres in meiosis 2, and chromosome segregation in meiosis 2 goes completely awry. Somehow you need both pathways to generate sufficient cohesion at centromeres that will persist after the first meiotic division and hold the dyads together during meiosis II. And you mutate either pathway, that cohesion is insufficient. Spores are basically spores in yeast. I presume also in you and me, but I'm not sure if that's been tested.
Good. So it's already 5:30, and I don't have more questions here on the screen. So with this, I really want to thank you, Kim. Fantastic seminar, and I hope that we can see you soon on campus. That we don't have you only virtually, but also can meet in real life and show you IST. So thanks again. It was really nice.
Michael, thank you very much indeed. It's a pleasure. I hope in the next few months, certainly sometime next year, I'll be in Vienna, and I'll let you know. And I'll come and visit. I don't know whether I have to give another talk, but people can ask me questions then.
Thank you very much. So keep enjoying the south of France.
Thank you very much, and take care, all of you.
Thank you, Kim. Thank you very much.
Not at all.